Hello and welcome to ThePrint. This is Akanksha Mishra and you're watching Scientifix, where I will be taking you through this week's top science news from across the world. Our first story today is about a new paper in Cell Reports journal which describes FGF21, a naturally occurring hormone that reversed obesity in mice. Scientists from the University of Oklahoma discovered FGF21, which is a hormone that's produced by the liver but it acts on the brain. In fact, the study found that this hormone acts on the same part of the brain that GLP-1 drugs like Ozempic do. In mice trials, the scientists found that this hormone reduced body weight and fat levels in blood by sending signals to the brain region that controls appetite and metabolism, which is again similar to Ozempic. However, there's one key difference between Ozempic and FGF21, which is the kind of reaction that they have in the body. So most GLP-1 drugs act as appetite suppressants, meaning that they act on the part of the brain which controls appetite and they increase the feeling of being full in people. But FGF21 works by increasing energy expenditure in the body, meaning that it basically helps the body burn more calories rather than reducing appetite. Our next story is the cover story of Nature journal last week. Researchers at NYU Langone Health have proposed a new model to explain how cancer cells become drug resistant. It isn't just through mutations, but by actively adapting. The study highlights AP1 proteins that are present in every cell, which help cells respond to stress such as chemotherapy. The study suggests that cancer cells use AP1 as a kind of trial and error system, switching genes on and off in different combinations to test survival strategies. Ineffective changes are discarded while successful ones are stabilized and remembered, allowing cells to lock in a resistant state and pass it on. This means that tumors can learn to survive treatment without changing their DNA. The finding challenges the long-held view that resistance is driven mainly by rare mutations. Instead, it points to cancer's flexibility as the real problem, and it suggests future therapies may need to block this adaptive ability to keep treatments for cancer working longer. Our next story is about a new discovery from Virginia Tech University. But it isn't by a paleontologist or a renowned scientist. Instead, it's by an undergraduate student. Simba Srivastava, a senior geosciences student, identified the fossil of Tychotheratus bucculentus, a 210 million year old dinosaur which is 3 times older than the Tyrannosaurus rex, which lived around 65 million years ago. It's not just the discovery that's interesting. Srivastava identified the dinosaur from a mangled, broken skull, which was dismissed as too deformed to be studied by scientists when it was first discovered back in 1892. But Srivastava spent 2 years carefully reconstructing the skull using CT scans and 3D printing, and he was finally able to identify that it was a new species of dinosaur from the Triassic period. Why this is important is also because there are currently very few fossils left from the Triassic period because dinosaurs weren't really dominant on the earth back then. Srivastava's study shows the skull's unusual features: massive cheekbones, a wide brain case, and a short snout, which tell us about the anatomy of early dinosaurs. Finally, researchers from the University of Copenhagen and the University of Illinois Urbana-Champaign conducted the largest ever genomic study of African elephants, showing how habitat loss is reshaping their genetic health. They analyzed 232 genomes from African elephants across 17 countries, confirming that both African savanna elephants and African forest elephants historically moved across continents, and they maintained genetic diversity through interbreeding. But in the present day, growing human pressures like agriculture and infrastructure have fragmented elephant habitats and isolated their populations. In regions like Eritrea and Ethiopia, elephants show clear signs of inbreeding and reduced genetic variation, which makes them more vulnerable to disease and environmental change. The study highlights that conserving elephants requires protecting not just the animals but also landscape connectivity. That's all we have for you today. Thank you for tuning into The Print.